Hello everyone, you are now watching TanyaCikgu live from BAC Fix Studio. My name is Fong and I will help you to answer all the questions regarding biology, SPM for next 2 hours. So you can send your question in the comment box and I will try my best to help you for the coming SPM. So at the moment, I think when we wait some of you to send the question, so I'm going to open one topic for everyone first. So for here, I'm going to discuss about the Mendel's formula so that this one, it will help you also in SPM. Because for the Mendel's formula, normally will come up 1 in paper 2. We can start with the Mendel first law, okay? So for the Mendel first law is about, we study only one pair of alleles. I give you example, let's say father is tall and mother is short, okay? Info here, father is tall and is homozygous, means double big T. And then the mother is short and is double small t. So when these two cross, you will get the gamete is just the big T. And for gamete, you have to circle. Based on the Mendel's formula, the gamete you have to circle. And then this one also the gamete, you have to circle also. When these two combine together, this is the fertilization process. And the result is one big T and one small t. So when it comes to all these points, if for the essay like this is not good enough to score, you have to write the title. Let's say the big T cross with the small t here. The title is genotype. It's about the DNA of parents. Then for the title gamete, you have to put the title there is gamete. And then for the offspring genotype, you write there genotype of offspring. When it comes to exam, you have to show all the topic here. Then only is considered correct. When it comes to the marking, it's very simple. Normally with the title, let's say genotype of parents, and the answer here is correct, you will get 1 mark. Then for the gamete, with the title and gamete, you got circle as well, you will get another 1 mark here. And the genotype for the offspring, answer correct, you get 1 mark here. And one more thing you have to write is the line here, if can, put the word meiosis to show that the process is actually meiosis. And then the line here is fertilization or random fertilization. So this one is actually for the Mendel first law. So the Mendel first law here, so this is the thing that you have to know. Okay, so we now start. Okay, so there's a question from Nurul Afifah. This is actually past year paper. Okay, so based on the diagram 2.1 and the 2.2, which is carrier protein, which is phospholipid bilayer. Okay, from the diagram, we can see that the one that open and then close, okay, open and close, that diagram based on that, the moving close one, that one is the carrier protein. Then of course for the phospholipid bilayer, you can see the phospholipid, the circle, and got 2 tail like this. And here also circle the 2 tail this one, you label the whole thing here for the phospholipid bilayer. Then the one that open and close, that one is the carrier protein. And based on this diagram, this actually Form 4 syllabus, Chapter 3. Okay, look at the diagram 2.1. You can see this one is the molecules from the top got more, bottom got less molecules. It means the thing moved from higher concentration gradient to lower concentration gradient. So for 2.1, this actually facilitated diffusion. And then for the bottom one, that diagram, you can see ATP involved. That one is the active transport. Okay, so come back to what you asked just now, which is carrier protein, is the one that open and close, can move that one is the carrier protein. And then for the phospholipid bilayer, you must label the whole thing, the phospholipid, the whole thing, and phospholipid bilayer. If you just point to one arrow to just the phospholipid, then it's wrong. Must be the whole thing for the phospholipid bilayer. I hope this one can help you. Okay, for the next one, okay, from this Soalan from Bone. So can you explain how cancer cells develop? Of course, yes. Okay, so about the cancer cell, this actually under Form 4 syllabus. Chapter 5 is under mitosis. Actually, cancer cell is under uncontrolled mitosis. Okay, you must understand how come cancer cell can happen. Number one, normally when come to exam, you have to know that the factor that trigger the cancer cell means the factor that can cause mutation. What can cause mutation? For example, UV, or maybe radiation, or any chemical that may trigger mutation. They are called mutagen, like benzene, CCl4. So this mutagen or maybe radiation, number one, they cause mutation. So first point is mutation. Mutation means the DNA change suddenly. It's mutation for the first point. Then the second point, from mutation may result in uncontrolled mitosis. Okay, uncontrolled mitosis. Then from uncontrolled mitosis means we divide very fast. Through mitosis very fast and faster than the normal rate. So it's called uncontrolled mitosis and form a group of cancer cell, means tumor. I repeat, the first point is you have to know the factor that trigger the cancer formation. For example, let's say it's radiation, or maybe it's UV, or maybe it can be any mutagen, any chemical that may trigger cancer. So that's number one. And then from here can cause mutation. So the mutagen cause the mutation. Then from the mutation, then form the cancer cell. Then the cancer cell here will perform uncontrolled mitosis, means very fast, and form a big size of the unwanted cell or tissue. So this unwanted cell here, a group of unwanted tissue, is called a tumor. Okay, yeah, so I hope that this one can help you. Just one more time here. Answer is mutation, uncontrolled mitosis, and form tumor. So normally about 3 points here. And of course, depends on the exam paper. If they show you is about the lungs cancer, then normally talk about smoking. That one is about TAR, T-A-R. That one trigger the mutation. If we talk about, let's say, other cancer, like skin cancer, normally caused by the UV light. Because from the sunlight, UV can cause the skin cancer. UV also can cause mutation. Is it okay or not? Okay, hope that this one can help you understand better about the cancer cell development. So it's under your Form 4 syllabus, Chapter 5, mitosis, application, uncontrolled mitosis. Okay, for the next one, okay, from this student called Navin. Okay, teacher, any tips to score paper 2? Okay, for this one, I give you the overall picture. For paper 2, how to target. I think this is a main concern for majority SPM student. Okay, for paper 2, you must understand the format first. You will have total 5 structure and 4 essay. Okay, for the 4 essay, we talk about this one later on, about the 5 structure. Okay, from the 5 structure, you can check from the SPM paper about almost every year. There are 3 structure questions that ask about the Form 4 syllabus. Then another 2 is about the Form 5 syllabus. So it's about 3 to 2, means 60% is about Form 4 and 20% is about Form 5. It means you have to focus more on your Form 4 syllabus to score more than half here. Okay, so for the structure, there are 3 structure Form 4. And from these 3 structure, normally got 2 structure actually come from Chapter 2 to Chapter 5. Like Chapter 2 is about cell, animal cell, plant cell. Okay, can talk about the organelles, can be also the cell organization, can also link to amoeba paramecium, Chapter 2. Chapter 3 is about the plasma membrane, the fluid mosaic model, and also the osmosis thing, simple diffusion, about transport, Chapter 3. Chapter 4 is about enzyme, okay, enzyme formation. The main one, lock and key, can be protein structure as well. And one more is the cell division mitosis, and meiosis. So these 4 very basic chapters for Form 4 will give you 2 structure. Normally got 2 structure come from these 4 chapters. Means you study chapter 2, 3, 4, and 5, you will get total 24 marks because one structure is about 12 marks. So 2 structure from these 4 chapter, of course they are random. Can be like chapter 2 and chapter 4 only come out, or chapter 2 and chapter 5 only come out, or chapter 2 and 3 combine as one structure, then chapter 5 alone another one structure. So total got 2 structure. So very standard for SPM, you study chapter 2, 3, 4, 5, you will get 2 structure. Okay, then another one structure about the Form 4 syllabus normally is chapter 6 or chapter 7. Chapter 6 is about nutrition, okay, the food intake, digestion, assimilation, or maybe it's photosynthesis. Then chapter 7 is about respiration, aerobic, anaerobic respiration, or can be human, can also go to the animal part. So chapter 6 and 7 is popular for paper 2. Normally we have one structure come out about chapter 6 or 7. Okay, so normally 6 or 7 we have one structure. If let's say come out is about chapter 6 for the structure, means chapter 7 normally is for essay. If the other way, if chapter 7 is structure, then chapter 6 is essay. So these two you study minimum one structure, or can be at least one essay also, or one structure, one essay. Okay, you can do your max calculation here. You study chapter 2 to chapter 5, you will get 2 structure. You study chapter 6 and chapter 7, you will get one structure plus one essay. Okay, then how about the last 2 chapters for Form 4, ecosystem, about the endangered ecosystem, about the pollution, and also the dynamic ecosystem. Normally ecosystem, the out is in essay. Yes, for the essay, ecosystem about the environment, about the pollution, the food chain, all this more to the essay. So we study the last 2 chapters, the one is for the essay. So, okay, one more time here. We have 5 structure altogether, normally 3 structure, Form 4, 2 structure, Form 5. So from these 3 structure, we have 2 structure here is from chapter 2 to chapter 5. Of course, it's random, okay? And sometimes you can mix the topic like chapter 2 and chapter 3 combined as one structure. Then chapter 5 come out alone one structure, okay? Then chapter 6 and 7 you study for one structure plus one essay. And then the essay, we have ecosystem as well. It's every year, ecosystem, pollution. Pollution can be like water pollution, eutrophication, can be deforestation, like last year and last 2 years, deforestation. Can also acid rain formation also. Okay, ozone layer depletion, more to environment, the one for essay 1. Okay, so it's like chapter 6 or 7, one for structure, one for essay, then one more essay is the ecosystem. All this for the Form 4 one. Then now we talk about the Form 5, move on to Form 5. So 2 structure from Form 5 and 2 essay from Form 5. Normally the 4 essay, 2 essay from Form 4, 2 essay from Form 5. So 2 essay plus 2 structure for the Form 5 one. Okay, the popular topic for Form 5. Number 1 is your Form 5 syllabus chapter 1, transport. Okay, for transport, last year out essay. 2 years ago also essay. Yes, this year might be essay or might be also the structure. So you can prepare for this year from chapter 1 transport, Form 5 syllabus, can be also the defense system. Okay, body defense mechanism, can be about the blood clotting mechanism. Blood clotting mechanism is also under defense. Number 2 is about the human heart structure, the loop, the blood flow, okay, or maybe can link to Soalan Keba, talk about the heart disease. Okay, heart disease is very common due to the fat, high cholesterol. Okay, this one is for chapter 1. And then another very popular for your paper 2 from Form 5 is your chapter 3. Means chapter 1, number 1, very popular. And then chapter 3. For the chapter 3 is about homeostasis, like how to control blood sugar level, how to control your body temperature also. And okay, what you need to know is chapter 3 got also reflex. So about the reflex action, how to explain reflex, okay? And then the hormone thing, and also the homeostasis for chapter 3. And then chapter 4 is about, okay, you can focus more on the baby formation, about the pregnant mother, because this one also popular. Chapter 4 is about reproduction. So about the pregnancy one, quite some time not come out already. You can focus more on the pregnancy, about the fetus formation, about the placenta thing, about the twins formation. We have the identical twins and the non-identical twins, and one more is the Siamese twins. Okay, then for chapter 5, Mendel. Normally Mendel can come out alone, or Mendel can pair with the chapter version combined in one essay or maybe one structure. Okay, Mendel's formula, you must know how to write the Mendel's formula, schematic diagram. Very important, and based on example given in the exam, but formula is about the same. Mendel will come out either Mendel 1 or Mendel 2, depends on that year SPM. So either 1 or 2, but it's still the same formula. Okay, okay, so at the moment is what we can do. Okay, so basically overall, you know the overall big picture, what you need to study, okay, for the SPM. Roughly, you know, okay, let's say my aim is just to pass the paper. For example, it's very simple. You study chapter all the form 4. All the form 4 alone is enough to pass. But let's say your aim is not to pass. Your aim is to score A or even A+, then no choice. You must put more effort and study everything. Okay? Okay, since now we got time, maybe I open another topic to discuss here. Okay, let's say we talk about body defense system. Okay, for human body defense system, we got the first line of defense, refer to our skin, and also the mucus. Okay, first line of defense, and actually blood clotting mechanism is also under first line of defense. So let's revise blood clotting mechanism first. Okay, when we get a cut, bleeding, we get injured. So once we get injured, okay, what happens is the blood will flow out, okay, bleeding here. At the same time, the platelet also will stick to the damaged tissue, about platelet. Then this platelet, once they stick at the damaged tissue, they will accumulate and form the one called platelet plaque. So from the platelet plaque, this form of platelet can produce actually an enzyme called thrombokinase. So, okay, here, one by one, when we get injured, the first blood cell that come to the injured area, answer is platelet. They will accumulate and clump together to form platelet plaque. Then from here, can release an enzyme called thrombokinase. Okay, this enzyme called thrombokinase got the ability to activate, or you can say to convert, a type of protein called prothrombin into the form called thrombin. Okay, what is the difference between prothrombin and just the word thrombin here? Answer is prothrombin is inactive form of protein, the blood protein, and then thrombin is the active form here. Okay, for the prothrombin here, prothrombin, this one is produced by the liver. And to form prothrombin in the liver, vitamin K is needed. Okay, once become active, of course, you want to activate prothrombin to thrombin, you need one core factor, is the calcium ion, to help the thrombokinase to convert prothrombin to thrombin. Once here become active, then the thrombin can activate or convert fibrinogen into fibrin fiber to stop the bleeding. Once fibrin fiber is formed, that's the end of the blood clotting. So in case you get injured, what happens is the bacteria can enter your body through the wound. So already like destroy the first line of defense. So we perform the blood clotting here, in which is seal it, bacteria cannot go in anymore. So actually blood clotting is our first line of defense. Okay. Okay, so we move on to the student question first. Okay, from another student, Marvin. Okay, based on the diagram first, okay, about the behavior of homologous chromosome, this actually crossing over. I can see from the diagram is about a crossing over process. And then they ask about which of the following is true about the product of homologous chromosome behavior, means about the crossing over the product. Okay, answer cannot be A. Why is not A is the answer, means why A is not the answer, because synapsis occurs, this one is before crossing over. So answer cannot be A. Then look at the answer B first. Exchange of genetic material occurs. Okay, this one B. And then replication of chromosome take place. This one no. Homologous chromosomes come together to form bivalent. Okay, so based on the A, B, C, D, the best option is B, because this diagram show you is the crossing over, means the two homologous chromosomes, they come together, and then at the touching point there is called the chiasma, and then they exchange the DNA. This is called crossing over. And the word synapsis, the answer A. Why not answer is A, right? Because when these two homologous chromosomes, they come together, they pair up. This process is called synapsis. So it should be after synapsis, then only got crossing over. You get what I mean or not? So it should be after pair up, after synapsis, then only got crossing over. So it's impossible the product of crossing over is synapsis. Means synapsis first, then only crossing over. Okay, this actually based on your syllabus, is your Form 4 syllabus, Chapter 5, under meiosis, is meiosis 1. This is actually the prophase 1. Okay, for prophase 1, you can recall, number 1, from the interphase to prophase 1, chromosomes condense, become shorter and thicker. Like from the diagram, you can see the X, one black color, and then one X, the white color. So these chromosomes, once they condense, they will pair up. So when they pair up, this is called actually the synapsis. The homologous chromosomes, one black, one white, one from father, one from mother, they will pair up. So once they pair up, this process is called the synapsis. After they pair up, they only at the crossing point there, this crossing point is called the chiasma. They exchange the DNA, and this is called crossing over. So basically, based on the diagram, I can see it's crossing over, so cannot happen synapsis 1, because after crossing over cannot be synapsis. Synapsis should be before crossing over. You get what I mean or not? Means synapsis first, they only crossing over. So the product cannot be synapsis again. It's before crossing over, got synapsis, means they pair up. Okay, yeah. I hope this one can help you. And look at the answer C as well. The replication of chromosome takes place. So replication, the one is for the interphase. So C cannot be the answer, because from one become the double. The one is called DNA replication, is for the S phase, means interphase. Then for the D, homologous chromosomes come together to form bivalent. That's the answer for synapsis process. So it's synapsis, they come together first, then only they exchange the DNA, means crossing over. Okay, yeah. Hope this one can help you to understand better about the difference between synapsis and crossing over. So pair up is called synapsis. Then only exchange the DNA is called crossing over. Okay, then we go to the next one. Okay, dalam bahasa, okay, boleh juga. Okay, Hakeem. Okay, ini adalah soalan bagi bab variasi untuk manusia. Okey, berdasarkan Raja 8.2, okey, menampak bahagian yang cubing telinga, bahagian yang ibu jari dan kebolehan untuk gulung lidah. You boleh gulung or you tak boleh gulung lidah. Okey, namakan jenis variasi dan terangkan puncanya. Okey, yang pertama untuk jenis variasi, okey, semua contoh di Raja 8.2 katakan you boleh ataupun tak boleh gulung lidah dikawal sepenuhnya oleh DNA kamu. Itulah faktor genetik. Kalau dikawal oleh faktor genetik sahaja, okey, untuk variasi, kita ada dua jenis variasi. Kamu tahu, yang pertama ialah variasi selanjah. Yang kedua ialah variasi tak selanjah. Apakah beza variasi selanjah dan variasi tak selanjah? Okey, untuk variasi selanjah, di mana semua contoh, okey, anda variasi selanjah dipengaruhi oleh faktor genetik dan juga faktor persekitaran. Sebagai contoh, katakan ketinggian kita. Ya, DNA kita boleh mengawal, tapi adanya faktor persekitaran seperti makanan. Makan cukup, adanya jin tinggi, makanan cukup boleh tumbuh tinggi. Itu adalah variasi selanjah. Tapi untuk yang diagram 8.2 ini adalah dikawal, dipengaruhi oleh faktor genetik sahaja. Jadi jawapan ialah variasi tak selanjah. Yang pertama ialah variasi tak selanjah. Namakan jenis variasi, jawapan ialah variasi tak selanjah. Boleh faham? Okey, kenapa ialah variasi tak selanjah? Katakan kebolehan untuk gulung lidah. You boleh gulung ataupun tak boleh gulung saja. Katakan, okey, saya adanya jin yang saya boleh gulung lidah saya. Mungkin tak? Lepas saya makan KFC, tak boleh gulung lagi. Tak mungkin kan? So, boleh gulung? Boleh gulung. DNA control punya. Boleh faham? Jadi, untuk kes begini, ialah variasi tak selanjah. Jawapan dia dan terangkan punca-puncanya. Bagi variasi tak selanjah, itulah faktor genetik. Dan ini ada untuk esei. Dia bagi 6 markah. You hanya beritahu, okay, namakan jenis variasi. Jawapan variasi tak selanjah hanya dapat 1 markah sahaja. Lagi 5 markah, you terangkan, okay, variasi tak selanjah ataupun contoh-contoh di sini dipengaruhi oleh faktor genetik sahaja. You dapat 1 markah. Then how to get another 4 points kan? Okey, jawapan ialah you kena terangkan. Okey, bagi faktor genetik ada beberapa punca punya. Katakan termasuk yang pertama ialah sinapsis. Ialah crossing over, iaitu ialah pindah silang. Okey, begini ya. Untuk bahagian faktor genetik ada lagi 4 punca yang boleh mempengaruhi genetik yang tak sama. Yang pertama, jawapan ialah pindah silang. Yang kedua ialah semasa kromosom homolog tersusun pada sata khatulistiwa, mereka boleh susun secara random. Boleh faham? Okay. Nombor 3 ialah random fertilization. Itulah persenyawaan rawak. Let's say when sperm ovum combine, boleh random juga. Dan last sekali, jawapan ialah mutasi. Tapi untuk kes ini, tak perlu terangkan mutasi sebab dia ialah kes yang normal. Normally mutasi untuk kes yang abnormal seperti sindrom Down, okey, yang sindrom Turner, yang abnormal case. Tapi untuk yang ni, you kena beritahu yang pertama, namakan jenis variasi, jawapan ialah variasi tak selanjah. Yang kedua, variasi tak selanjah dipengaruhi oleh faktor genetik sahaja. Lepas tu, daripada sini, faktor genetik terangkan lebih lagi. Okay, punca-punca under faktor genetik termasuk pindah silang. From pindah silang, terangkan lebih lagi. Pindah silang, 1 markah. Okay, terangkan apa itulah pindah silang. Pertukaran bahan genetik antara kromosom homolog. Dan apakah akibat dia? Menghasilkan variasi dalam gamet. Jadi tak sama DNA. Boleh faham? Dan terangkan lagi punca lain, katakan ialah semasa persenyawaan, sperma dan ovum digabung secara random. Maksudnya bukannya jawapan yang tetap. Okey, sebagai contoh, okay, bayangkan saja, katakan saja, okey. Bandingkan kamu dengan kakak kamu ataupun kamu dengan abang ke adik kamu. They say daripada satu keluarga di mana mungkin some of you boleh gulung lidah. Some of your abang adik tak boleh. Kenapa? Sebab ialah masalah DNA sahaja. Boleh faham? Sebab manusia kita ada kebolehan boleh gulung ataupun tak boleh gulung ialah DNA control. Okey. Kalau tanya saya, mungkin tak dua orang yang boleh gulung lidah menghasilkan anak yang tak boleh gulung lidah? Memang boleh. Memang boleh ya. Katakan kebolehan untuk gulung lidah dikawal oleh gene dominant. Okey, dominant gene, gene dominant. Okay, katakan. Ini already ada sikit out topic ya, just bagi extra saja. Katakan kalau seorang dia boleh gulung lidah dengan keadaan ialah heterozygous. Katakan ni ayah dan ibu. Dia pun boleh gulung lidah, G ialah gulung. Okey, ini pun hetero. Jadi untuk mereka mungkin tak dapat anak yang tak boleh gulung lidah mereka? Memang mungkin. Sebab satu recessive G daripada ayah dan lagi satu recessive G daripada ibu. So double small G jawapannya tak boleh gulung. How about dua-dua ibu ayah pun tak boleh gulung? Jawapan hanyalah tak boleh gulung sahaja. Tapi untuk sebelah sini pun mungkin boleh gulung juga. So ada dua jawapan. Dan semua ini dikawal oleh faktor genetik sahaja. So untuk faktor genetik, punca-puncanya terangkan lebih sikit dari segi pindah silang, semasa persenyawaan secara random dan semasa kromosom homolog tersusun pada sata khatulistiwa also secara random juga. Okey, jadi sini cukup poin untuk 6 markah. Saya ulang. Yang pertama, namakan jenis variasi tak selanjar. Terangkan lebih sikit, dipengaruhi oleh faktor genetik sahaja. Daripada sini, terangkan lebih lagi. Termasuk pindah silang. Pindah silang, terangkan lebih bagi pindah silang. Apa itu ialah pindah silang? Pertukaran bahan genetik antara kromosom homolog. Apakah kebaikan menghasilkan variasi? Lepas tu, yang kedua, semasa metafasa 1. Okey, metafasa 1. Okey. Kromosom homolog tersusun pada sata khatulistiwa. Kat tengah-tengah sekali, secara random juga. Boleh faham? Ini pun boleh menghasilkan variasi. Ini sama dengan Mendel hukum nombor 2. Lepas tu, lagi satu ialah semasa persenyawaan berlaku secara random. Katakan, ibu punya sel telur, ayah punya sperm, mereka boleh gabung secara random ikut hukum Mendel. So boleh menghasilkan banyak jenis phenotype yang tak sama. So adanya variasi. Okey, so ini soalan dia. Harap ini boleh membantu kamu memahami lebih tentang variasi. Okey, next question. Okey, ini pada student namanya Kat. Okay, for paper 2 essay, is it better to write in point form or paragraph form? Okay, I think for this point, a lot of argument from school teacher and also some teacher also, we should write in paragraph form for the essay or should be in point form. Some they say we should write in point form. Some they say we cannot write in point form. Exam will deduct your mark. Actually, point form, you can write in point form. But the point is, even in point form, you must write everything in complete sentence. Even in point form, you can write in point form, but your answer is in complete sentence. Not just one keyword, then you full stop. Other one cannot. Because those days got some student, they think, oh, I can write in point form, means I give only the keyword. I don't want to explain anything. Just a keyword, and keyword, just short answer, and short answer. That one you cannot score because it's still essay. You want to write in point form? Can. With the condition, you write everything in complete sentence plus your keyword in. Then you can score. Actually, when you put in point form, all the point form, every single point in complete sentence, you just combine them. You can put also in paragraph form. So they are the same thing. Just that, okay, you write everything in paragraph, compared to write the point form one by one, but all in complete sentence. actually you write the same thing, just that the way you present your answer in point form or in paragraph form. So both also can for essay, don't worry, okay? But of course, the point form, just the keyword, one keyword, then you cannot score. Okay, let's say, I want to explain about the osmosis. The condition is one cell immersed in the hypertonic solution. There you can see the diagram, the membrane, let's say for the plant cell, the membrane move away from the cell wall. Correct or not? The plant cell immersed in, let's say, very concentrated salt solution. Water diffuse out. I can see the membrane move away from the cell wall, means answer is plasmolysis. Let's say for this one is in essay, I just write number 1, hypertonic solution, full stop. Number 2, water diffuse out, full stop. Number 3, plasmolysis, full stop. Like this is wrong. You have to write in complete sentence. For example, number 1, you have to mention the concentrated salt solution is hypertonic to the plant cell. Full sentence, you can score. Compared to the word hypertonic, full stop, the one cannot score. Understand? So don't worry, when it comes to exam, you can put in point form also can, paragraph form also can, as long as it's complete sentence, it's still essay. Okay, hope we can help you. Okay, next question is from Enzin. Why the answer is C? Okay, it's from SPM 2018 from the past year. Okay, based on the diagram here, it's about the leaf structure. Gas X is used in a process that uses light energy. Means this gas must be the carbon dioxide, because carbon dioxide is used in photosynthesis. We talk about carbon dioxide here. Okay, which of the following is correct regarding gas X at the compensation point? Okay, look at your answer C first. No net gain or loss of gas X at compensation point. And then for the answer D, okay, loss and then gain of gas X. Okay, so why answer is C? Answer C is the correct answer. Okay, you must understand number 1, this gas is actually carbon dioxide. Okay, and then another keyword is at the compensation point. What do you mean by compensation point? It means the rate of photosynthesis same as the rate of respiration. So when plant perform photosynthesis and respiration at the same rate, so all the carbon dioxide that released by the mitochondria, all will be used by the chloroplast to perform photosynthesis. So no CO2 diffuse out. Okay, just nice all the CO2 supply to the mitochondria to perform respiration. Then from here, when they produce the oxygen, all will be used also by the, when here produce, another way, respiration, respiration produce carbon dioxide, then all is used by the chloroplast to perform photosynthesis. Then from photosynthesis, release the oxygen. Okay, and all used by the mitochondria for respiration. So the other way, okay, one more time here. For the gas X is carbon dioxide. At the compensation point, respiration happen at the same rate as the photosynthesis. So they happen at the same rate. It means all the CO2 gas that released by the mitochondria, let's say here release total 10 CO2, let's say, molecule, all will be used by chloroplast for photosynthesis. Chloroplast. Of course, from here, they will release oxygen. From photo, also release 10, let's say. They all will be used by the mitochondria when come to compensation point. Even the respiration produced total is 10 CO2, all will be used by the photo, and here produce also 10 oxygen. So it's a cycle. Compensation point normally happen early in the morning when there's a little bit sunlight and the rate of photosynthesis equal to the rate of respiration. At this point, there's no gas exchange. Even the stoma open, daytime stoma open, but when it comes to compensation point, there's no gas exchange. So there's no loss or gain of the carbon dioxide as well as oxygen. Of course, back to here, gas X, I can see answer is actually carbon dioxide. So for compensation point, there is no lose or gain carbon dioxide because the gas just circulate within mitochondria and the chloroplast within the mesophyll cell, let's say. Okay, so answer C is actually correct. Okay, I hope we can help you. Okay, next one from the same student. Okay, what is epizoid and the epiphyte? Okay, epizoid, the zoid actually for animal. Okay, for animal. Epiphyte, the phyte is actually for the plant. So epizoid and epiphyte, you can get this term is your Form 4 syllabus about the symbiosis interaction is under commensalism. So for commensalism, we got one is called epizoid, means it's under symbiosis. Under symbiosis, we got one interaction is called commensalism. Okay, commensalism. And then under commensalism, if the interaction between two animal, then it's called epizoid. Between two plant, it's called epiphyte. For example, let's say for the epizoid, common example is remora fish. Always swim under the big one fish, shark. So the remora actually want to get free protection from the big one, the shark. But for the big one, there's no harm, also no benefit, but remora benefit. This is called commensalism. So this type of commensalism, talk about animal one more term is called epizoid. Zoid refer to the animal. And then for the epiphyte, let's say in forest, got one tall tree, and then got the small plant like orchid grow on the big tree. So this small plant grow on the big tree, actually they can perform their own photosynthesis. They don't absorb any nutrient from the big tree. So basically for the big tree, it's no harm. Of course, also no benefit. But for the small plant, benefit. Because can get higher position in forest to absorb more sunlight. So one benefit, one remain the same, is also commensalism. But about plant, one more name is called epiphyte. Okay, I repeat here. So epizoid and epiphyte, both also under commensalism interaction. Just that epizoid, the zoid refer to commensalism between animal and animal, the interaction. And then epiphyte is actually for the plant and the plant interaction, both also commensalism. I hope that it can help you to understand better. Yes, it's very close. Epizoid, animal, epiphyte for plant. Okay. Okay, from the same student. Okay, it's also past year, so also 2008 here. Okay, why the answer is A? Okay, go to first. Which genetic disorders are caused by a sex-linked gene? Okay, so based on syllabus for the sex-linked gene, okay, we only study two. One is actually the hemophilia. Okay, hemophilia. And then number two is the color blindness. So 1, 2 is already confirmed one. Okay, 1 and 2 is already confirmed one. Okay, and then for the muscular dystrophy, this one is not in the chapter Mendel, but it's in another chapter. Muscular dystrophy is also under sex-linked gene because muscular dystrophy normally attack male. Normally will affect is the male. If normally affect male more than the female, it means the gene is located on the X chromosome. Okay, one more time here. For the thalassemia, this one is actually about the normal gene only. Okay, now talk about the sex-linked gene. Sex-linked gene means the gene located on the sex chromosome. If located on the sex chromosome, normally it's located on the X chromosome. For girl, XX. For boy, is the XY. So normally this sex-linked gene, is the DNA on the X chromosome. Based on our syllabus, we studied the Form 5 syllabus about the Mendel, the sex-linked gene. We studied 2 examples. One is hemophilia, confirmed already. Number 2 is color blindness, confirmed already under sex-linked gene. Another one, the muscular dystrophy, is it in our syllabus? Answer is yes. But they never explain about the muscular dystrophy located on the sex-linked gene for chapter 5. They explain this one more to chapter 2. Form 5, chapter 2. Form 5, yes, chapter 2 about the locomotion and support. The last part there got the muscular dystrophy one. Okay, then from there, they got explain actually muscular dystrophy is under sex-linked gene. Muscular dystrophy means the muscle degenerate. Like the leg muscle becomes smaller, weaker and weaker. So these people normally they are on wheelchair. They are very young, but they are on wheelchair and normally is male because muscular dystrophy is the gene located on the X chromosome. So it's inherited. Okay, so answer A is actually correct. Okay, I think your main point is since you circle D, you can understand also 1 and 2 confirm correct. Just that 3 and 4 only. That's the part that you feel a bit confused. So just to clarify, muscular dystrophy here, yes, it's the sex-linked gene. So normally attack male more than female. Okay, yeah. So about the percentage, 50-50. For male, okay, next one first. Okay. Okay, can I explain about the difference between gene mutations and chromosomal mutations? Okay, this one, yes, sure. Okay, for the gene mutation, only one gene mutate. Okay, let's say we got 46 chromosome. Okay, if the number of chromosome remain the same, still 46, every chromosome also still perfect, healthy one. But within one chromosome, there's a gene that mutate. Only one gene mutate is called gene mutation. Okay, for example, let's say this one is one chromosome. One chromosome. Total, we still got 46 chromosome total, still everything the same. But talk about one chromosome, this is the gene. Let's say this gene is to control the black pigment formation. If only this gene here mutate, after mutation, of course, you cannot form the black pigment anymore. Then the disease here is called albinism. Albinism is under gene mutation. Or when come to exam that time, you can see the info, let's say from here, this gene got the DNA is the ATC, let's say, and then GTT, and then CCC. Let's say after mutation, the nucleotide now changed to ATC, remain the same, no difference. From here, GTT changed to GT. Okay, the last T become A, is GTA. And then here CCC remain. So which part mutate? The T here become the A. So only change one base pair. Only change one nucleotide only. So this one is called gene mutation. Okay, only one gene that mutate. But for chromosomal mutation is something more serious. Okay, for chromosomal mutation, got two possible factors. One is abnormal number of chromosome. Abnormal number of chromosome can be like 45 chromosome or 47 chromosome, because normal should be 46 for human. For those with 45 chromosome, example, is 44 autosome plus 1X. Lack of one sex chromosome is 45 in total, abnormal, or 47, 44 plus the X XY. Here extra one sex chromosome or 45 autosome means for the Down syndrome. This one is for the Down syndrome. And here can be girl, double X, or can be boy, XY here. So basically, if got 45 or 47 chromosome, is something abnormal here. So involved is the chromosomal mutation. Okay, yeah. Number 2 is the abnormal structure of the chromosome. Okay, let's say this is the normal chromosome structure, butterfly. After mutation, the chromosome here become, let's say, shorter. Something missing here. Can you see here? Something missing, the gene missing. And then here all normal. So if you got something missing, the whole gene delete already. So this one is called, actually it's called deletion. Delete the gene. Okay, let's say this is the gene, example, they can control your finger formation. So the gene disappear here. So the finger cannot be formed, or certain fingers cannot be formed here. So this one, the whole structure here mutate. Answer is chromosomal mutation. So in summary, one more time. Okay, for the chromosomal mutation, involve abnormal number of chromosome. Normal is 46. So for this abnormal number can be 45, can be 47, means a lack of one or maybe extra one chromosome. So it's under abnormal number of chromosome, it's under chromosomal mutation. Okay, I give you an example. So normally under the abnormal number, the most popular for exam is the Down syndrome. Down syndrome got one more name, it's called trisomy 21. So the extra one chromosome is on pair 21. So extra chromosome. Then number 2 is the Klinefelter XXY. Number 3 is the Turner, only 1X. So short of one chromosome. Okay, yeah. So from here, all this abnormal number is under chromosomal. All the syndrome is very serious case. And abnormal structure also very serious. Let's say the gene to form, example, this finger is missing. So this finger I cannot have, I cannot get. Okay, because the gene is missing. You get what I mean here? So it's under chromosomal mutation. But for the gene, everything is still perfect. Only one gene cannot perform. Let's say if the gene is to control the black pigment formation, what happens is, okay, if the gene mutate, the DNA sequence of the gene mutate, the ATCG, the one mutate is gene mutation. Only no black pigment, so the hair, everything is no more black, no more dark skin here, the whole thing will be white or brown. That one is the gene mutation, means the albinism, albino. Okay, okay. Beside albinism, sickle cell anemia, okay, sickle cell anemia, and also the hemophilia, all this under the gene mutation. Okay, let's say, let you compare. Do you think anemia is more serious? Anemia is under gene mutation. Is more serious or Down syndrome? 47 chromosome. Down syndrome is under chromosomal mutation. Do you think Down syndrome is more serious or hemophilia disease is more serious under gene? Of course, it's the chromosomal one. Understand? So when talk about the chromosomal, either is the whole chromosome disappear, so 45 chromosome, or you get extra one whole chromosome, become 47, or the chromosome structure suddenly become shorter or longer. The structure change completely. Then it's also chromosomal. And then for the gene, everything still perfect. Number of chromosome is 46, perfect. The structure of the chromosome, no shorter, no longer, everything, the shape still perfect. But only one gene, the DNA inside the gene mutate. The ATCG change a little bit, but everything still okay one. You get what I mean or not? So let's say for hemophilia, only the blood clotting cannot perform, but the whole body is perfect. Okay, it's under the gene mutation. So yes, this one is actually is a good question because can come out also in essay or in structure. As you explain the differences between gene and chromosomal. Okay, so hope it can help you. Okay, now come from another student. Okay, Nadarajan, explain about the light reaction and dark reaction. Okay, so this one is a bit long story about the light and dark reaction. Should be a hot topic for this year also, because for quite some years not come already. Okay, so for the light and dark reaction can come out also in essay. If essay you have to explain from A to Z already. Okay, from the light reaction, every single step, to the dark reaction, everything until sugar is formed. And then for the structure, if come out in the structure, normally light and dark reaction can also link to Soalan kebat. But we come to the basic, I explain to you first about the light and dark reaction, let's say for the essay format, then you come to the Soalan kebat. Okay, yeah. Okay, for the light reaction and the dark reaction, first thing you have to know is about the equation for photosynthesis. They need carbon dioxide, they need water in the presence of sunlight, and also chlorophyll. Here must say chlorophyll, must be chlorophyll, not chloroplast. And from here, product, main product is glucose, and also the waste product or the byproduct is oxygen. About the balance part, don't worry, because it's essay. Okay, it's essay, no need to balance one. Okay, for step number 1, sunlight energy is absorbed by chlorophyll. You get 1 point. Means under the light reaction. Now we talk about light reaction. Tindak balas cahaya. Light reaction. Okay, in the light reaction number 1, light reaction occurs in grana. Grana, granum also can. You mention the site for essay, you get also 1 point. In the light reaction, sunlight is absorbed by chlorophyll, you get 1 point. Sunlight energy is used to split water molecules into hydrogen ion and hydroxyl ion. This one is another 1 point. And this step is also known as photolysis of water. So mention photolysis of water, here you score another 1 point. Okay, so now recap. Number 1 is light reaction occurs in grana. You get here 1 point. In light reaction, sunlight is absorbed or is trapped by chlorophyll, point number 2. And then sunlight energy that absorbed by chlorophyll is used to split water molecule into hydrogen ion and hydroxyl ion. You get point number 3. And this reaction is also known as photolysis of water, point number 4. So you get 4 points until here. Then we continue the next one. For the light reaction, this hydrogen ion will accept electron from, who gives the electron? From the chlorophyll, to become hydrogen atom. And then from here, the hydrogen atom will come out from the grana, move to the stroma, react with the CO2. Of course, when here react, this is actually the dark reaction. This is step 2. Just now the light reaction is step number 1. Okay, okay, so one more time here. Just now already got 4 points, right? Until this one we stop. So next one is hydrogen ion accept electrons from chlorophyll to become hydrogen atoms. Here you get 2 points. Because you mentioned the H plus accept electron, become hydrogen atom, you get 1 point. You mentioned the electron is come from chlorophyll, you get another 1 point. So got 2 points altogether. Okay. So just now got 4 points plus here 2 points. Total now got 6 points from the light reaction. Actually, light reaction, you can add more points from this side. The hydroxyl ion here will actually donate electron to chlorophyll to become hydroxyl group. Here can get extra point 1. Then the hydroxyl groups combine to form oxygen, the same oxygen, and also water. This is extra 1 point. Okay, one more time. From the hydroxyl ion, donate electrons to chlorophyll, 1 point. Then to become hydroxyl groups, 1 point. Then hydroxyl groups here combine, means many hydroxyl groups combine to form oxygen and water, is also 1 point. So here another 1, 2, 3 points already. All this from the light reaction. So total light reaction got 9 points. Okay, now we move on to the dark reaction. Okay, for the dark reaction, here, dark reaction, hydrogen, okay, sorry, first to mention, dark reaction occurs in stroma. Stroma, you get here 1 point. Then, in the dark reaction, hydrogen atoms are used to fix. Fix means react. Fix carbon dioxide, you get 1 point. To form glucose, actually, and water. Here got water, here goes water. Okay, one more time. Dark reaction occurs in stroma, is 1 point. In the dark reaction, hydrogen atoms are used to fix carbon dioxide, 1 point, 2 points already. And then to form glucose and water, this is also 1 point. So for the dark reaction, you only can get 3 points altogether. So light reaction, dark reaction, here total 12 points. So even for the essay 10 points, you can write any 10 points from here for the essay part. Okay, how about when they come out, it's not for the essay. If let's say for the structure, if for the structure, they will ask about, okay, maybe just explain the light reaction, or just explain the dark reaction, give you about 3 marks to maximum 5 marks maximum, okay, for the light reaction for structure. And then from here, can link to also the Soalan kebat. From the photosynthesis, how to link to Soalan kebat? Okay, for photosynthesis, they need light. They need also the carbon dioxide one. Okay, so if let's say the environment cannot provide enough sunlight, then the rate of photosynthesis will actually drop. Correct or not? Okay, let's say there's one Soalan kebat from the past year paper. When plants, they are planted nearby the factory area. Okay, explain how the plant grow, about the photosynthesis one. Answer is, the rate of photosynthesis will decrease. Why will decrease? Because factories is very dirty, dusty, so we block actually some of the light coming. So with lower light intensity, lower rate of photosynthesis, number 1. Number 2, the dust may cover the leaf surface, block the stomata for gaseous exchange. So if lesser gaseous exchange means absorb less carbon dioxide, then less photosynthesis. Then here is about Soalan kebat one. Okay, how about like this? It talk about global warming, greenhouse effect, increase in the CO2 in the atmosphere. Can this one help in photosynthesis? When I say CO2 gas increase, let's say from burning, okay, about pollution part, burning, then release more carbon dioxide. This carbon dioxide help in photosynthesis. Okay, so with more CO2, the plant can perform more photosynthesis. So for plant, it's actually a good thing. But when the temperature increases to very high temperature, the enzyme in the plants will be denatured. So once denatured, the plant will die. Cannot perform photosynthesis anymore. Actually, photosynthesis also involves enzymes. So at first, increased temperature is good, but too high, then it's not good for the plant because enzyme will be denatured. Okay, hope this one can help you. So you must understand, number 1 is the overall picture, and you have to compare also. Like if they ask you about the difference between light and dark reaction, then how to answer? You have to mention that light reaction requires light. Dark reaction does not require light. Light reaction occurs in grana. Dark reaction occurs in stroma. Okay, so it's like different site in the chloroplast. And also they require different things. Light reaction, they need actually water. Dark reaction, they need carbon dioxide as the raw material. Okay, hope this one can help you. Okay, from the next one, from another student. Oh, Jesse Hope. Okay, special, the name. Okay, how to memorize the nervous system. Okay, you must understand nervous system for SPM level. They will only ask you a very basic thing about the human nervous system. For example, about the afferent neuron, about the efferent neuron, and also the interneuron. Okay, about the neuron thing. Okay, so it's about the afferent neuron, efferent neuron, and the interneuron. So normally it's about the diagram. Let's say we talk about reflex action. For example, exam give you one diagram. It's about the spinal cord. The spinal cord cross section. You will see the gray matter at the middle, and then the surrounding is actually the white matter. And then from here, we link all the way to the spinal nerve and then to outside. So maybe ask you to draw, okay? From the receptor all the way can link to the spinal cord is called the afferent neuron, the input. Input is the afferent. So for the afferent neuron, one more name is called the sensory neuron. Why afferent neuron? Another name is called sensory neuron because of the neuron receives the impulse from our sensory receptor. Whatever we detect, okay, let's say even I close my eye, I feel something like this. I can actually feel it because I got the receptor. So from the receptor, it will send the impulse all the way to the spinal cord and then to the brain. That I know that I'm touching something hard surface, for example, because here got the pressure receptor. So we send impulse by the afferent or the sensory neuron. So once the impulse come to the spinal cord, the CNS, the central nervous system, in the spinal cord and the brain, the impulse is passed to the interneuron. Okay, interneuron is very special. Only for the brain and also the spinal cord, we have the interneuron. So this interneuron will send impulse all the way from the spinal cord, all the way up to the brain to let you know what happened outside. Okay, and then here we'll process the data and send back to the spinal cord. So everything up and down is the interneuron. And then from the interneuron in the spinal cord, we'll pass the impulse to the motor neuron, the efferent, and then for you to respond. Okay, let's say we talk about reflex. Let's imagine this table is extremely hot plate, hot table here, the surface here. So if my hand, let's say, accidentally touch a very hot object, I will like withdraw my hand immediately. This is called reflex. And for reflex, is controlled by the spinal cord. So you must understand, from I touch, I will have, of course, I have the receptor here, right? I have the receptor here. It's called the hot receptor or the pain receptor. So from here, trigger the impulse. Impulse is carried by the afferent neuron all the way sent to the spinal cord, the interneuron. And then from here, the impulse will pass to the interneuron in the spinal cord. From the interneuron, pass the impulse to the motor neuron and send to the muscle for me to withdraw my hand from the hot object. Because reflex, the main purpose actually is to protect your body from serious injury. Okay, about the structure, I give you an example. We talk about reflex. Let's say exam give you this diagram. Spinal cord, cross section. This is the gray matter. This is the white matter. Jirim putih. Will be the same thing here, left and right. Here is the spinal cord. Okay. So let's say here got the receptor. From you detect a very hot object or very sharp object, let's say. So from the receptor, the impulse will be carried by the sensory neuron. Okay, here's a sensory neuron. All the way go in. and until the gray matter, then you stop. For the ending, you draw like this. This one is really the axon terminal. And this swollen part here is for you to draw the cell body, the nucleus here. This one is the afferent or sensory neuron. And then, of course, in the gray matter here, you draw one more circle, the dot, the cell body, and all the way down here, must be within the gray matter. This one is the interneuron. This is the interneuron. And then the motor is here, circle, dot, and then all the way out to the effector, here in, here out. This is to the muscle to respond. Okay, this one is the efferent neuron. One more name for efferent is called the motor neuron. So basically, you can see from the diagram, okay, for the afferent neuron, the cell body is here. Can you see or not? Efferent neuron, the cell body is in the gray matter. So when you see from diagram, you can see the cell body is along the axon. So basically, that one is for the sensory. If the cell body is at the end or in the gray matter, the circle one is inside the gray matter, inside the spinal cord, this one is actually for the efferent neuron. So along the axon, cell body, the nucleus at the middle there, the one is for the interneuron. Okay, so this is about the neuron thing. If we talk about the neuron system, or maybe other word is the nervous system. So for the nervous system, you have to memorize the thing is about the brain. Because for nervous system, we got the CNS and the PNS, right? So the CNS is about the brain one. For human brain, there are few main parts. We have the cerebrum. We have behind here is called cerebellum. And here is called the medulla oblongata. There are three main parts. We have cerebrum. The floated layer here is called the cerebrum. The behind here is called the cerebellum. And just above the spinal cord is called the medulla oblongata. In terms of function, a slightly different function. Let's say we have emotion. We got the ability to learn new things, right? So basically, it's a cerebrum control. Here can control all the voluntary action as well. Our IQ, our memory, action that we can do, everything is cerebrum control. This is the main control center here. Then for the behind one, it's called cerebellum. I want a cerebrum, brum. This is cerebellum, belum lagi. So the cerebellum one is to control balancing. And then for the medulla oblongata, that one is actually to control our involuntary action, like your heartbeat. You're saying, I never control my heart to beat faster or slower by my will. It's just beat like that. It's automatic. So something automatic is called involuntary action, is controlled by the medulla oblongata. Okay, you feel like, okay, you want to, suddenly you feel like you want to vomit, just automatic. Those also involuntary, is medulla control. So basically, okay, one more time here, for cerebrum is for voluntary, memory, learning ability, behind called cerebellum, balancing, walking, standing, okay, body balance, and the medulla oblongata is to control all the involuntary one. Okay, okay, hope this one can help you. So basically, you have to memorize is about the brain, the structure and function, and also for this one, the diagram, afferent, efferent, and the interneuron. Okay, and about the function, they are all the same function. They are nerve, they are neuron. Only got one function for all this is to transmit nerve impulses. Means to carry nerve impulses. But transmit is better term. Okay, for bio, transmit impulse better than carry impulse. Same one. Okay, and of course, got a little bit different is the distance. Okay. Okay, for next one. Okay, from another student. Okay, Cikgu Fong, bagaimana ubat tahan sakit membantu pesakit di hospital? Is it same like drugs? Inilah tentang medical already ya. Okay, ubat tahan sakit means the painkiller. Okay, ubat tahan sakit. Okay, Kalau kamu masih boleh ingat, di bahagian neurotransmitter kan, kita adanya bahan kimia, neurotransmitter, antara satu saraf dengan the next saraf, di mana itu gap dinamakan ialah synapse. Lepas tu, synapse di bahagian dendrik adanya banyak reseptor kan, di mana reseptor ini dia boleh terima neurotransmitter. Okay, painkiller ialah block di bahagian receptor. Let's say this one is for the pain receptor. From the pain receptor, the impulse all the way sent to the next nerve is about the pain feeling, sakit punya. So ini painkiller, they will actually block the receptor. When they block the receptor, katakan contoh ini benda yang tajam, contohlah cucuk sini, sakit kan? Sebab impulse akan dihantar ke bahagian otak. So saya akan rasa sakit. But from I cucuk sini, impulse yang pertama dibawa oleh afferent neuron sampai ke interneuron. Interneuron hantar ke bahagian otak kan. Okay. Antara satu neuron dengan lagi satu neuron, antaranya ada satu ruang. Ruang ini dinamakan ialah synapse. Dan di ruang ini adanya bahan kimia yang dinamakan ialah neurotransmitter. So the painkiller can actually, okay, let's say this receptor can receive the bahan kimia from the neuron before this one. The painkiller can block here. Once they block here, the impulse tak boleh sampai. Kalau impulse tak boleh sampai, you tak akan rasa sakit. So normally untuk painkiller is about the neurotransmitter, bahagian synapse, because they block actually the receptor that receive the chemical untuk rasa sakit punya. They block there, so you don't feel the pain. Boleh faham? Okay. Untuk drugs, of course ada beza. Drugs ada banyak jenis. We got some is speed up the impulse or slow down the impulse. For painkiller is to slow down the impulse so that you tak rasa begitu sakit. Okay. Okay, hope it can help you. Okay, next question. Okay, can I explain about blood pressure and I think should be perkataan nephron. Okay, blood pressure and nephron. So it's one by one. Okay, blood pressure dulu. Okay, untuk blood pressure. Okay, yang ni ada di bahagian ialah tingkatan 5, form 5. Form 5 bab 1. Form 5 bab 1, because the receptor here is called the baroreceptor, is to detect the blood pressure. Okay, about the blood pressure actually is not too complicated. Okay, number one, let's imagine when you do exercise, your heart will pump faster. When your heart pump faster, the pressure of course become higher. So this high pressure during exercise is good thing. But once you rest, your body no need so much blood already, but the pressure is still a bit fast, right? At first, when you stop after running, you stop, the heart will slowly back to normal, slowly recover one. Because once you slow down, your body no need so much blood already, means sugar and oxygen. So your brain will let you adjust back to the normal pressure. Okay, let's say when your pressure is a bit too high already, number one, this high blood pressure will be detected by the receptor is called baroreceptor. Number one is the baroreceptor. Then from here, this receptor is stimulated and send impulse to the brain. So which part of the brain control your blood pressure? Of course, it's automatic, so it's medulla oblongata. So how to inform from the receptor, inform the brain is carried by the afferent neuron. Okay, it's by the afferent neuron here, send all the way to inform the medulla. Okay, say that now pressure is too high. Okay, when the pressure is too high, your medulla oblongata got the control center will adjust your blood pressure according to your body need. So this impulse will all the way send to the heart muscle, of course by the efferent neuron, because input is the afferent, output is the efferent neuron, send about the impulse. So basically, now pressure is too high already for your body need. So you want to lower the pressure, right? So what happened is, number one, the high blood pressure is detected by baroreceptor, B-A-R-O, baroreceptor. Then this receptor is triggered and send impulse to the medulla oblongata by the afferent neuron. And then from here, your medulla will adjust. This is a control center. Then from here, it will send impulse to your body to adjust. send impulse to your heart so that the heart beat slower. Okay, number one. Number two, breathing also back to normal, because when the heart beats slower, automatic pressure will drop. Then once back to the normal level already, it just everything continue normal, means recover already. Okay, in another way is, how about when the pressure is too low, just terbalik. Okay, yeah. When the pressure is too low, you need more blood pressure, you need more blood supply to the whole body, you have to increase the blood pressure. How to increase the blood pressure? Send impulse to the heart to beat faster. Okay, so one more time here. When your body pressure, the blood pressure is too high, make it lower. How to make it lower? Heartbeat slower. Okay, when the pressure is too low, make it increase the pressure, heartbeat faster. But to explain this one, always start from the receptor, is baroreceptor. And then send impulse to the control center, is the medulla oblongata. And then input must be the afferent neuron. Then output must be the efferent neuron to effector to respond. So for this case, the main effector is about the heart. Are the heartbeat faster or slower? Of course, even to explain more for essay, is about the blood vessel. Blood vessel dilate or constrict also can affect the pressure one. Okay, then we move on to the next one, is about the nephron. So nephron is about urine formation. Okay, nephron here. Okay, nephron is about the urine formation here. So for urine formation, okay, like this, you will have this structure, the cup-like structure called Bowman's capsule, proximal, loop of Henle, and distal. Okay, this is the nephron, for the nephron structure here. When it comes to this structure, the popular part is you need to explain the process that occur between the glomerulus. This is the glomerulus. Sorry. Glomerulus. And this is the Bowman's capsule. Okay, name the process that occurs between glomerulus and the Bowman's capsule here. So answer is called ultrafiltration. And then you explain or describe ultrafiltration. First, you must mention the high pressure. But this high blood pressure, you change to high hydrostatic pressure. This high hydrostatic pressure will actually in the glomerulus one and force some blood components out into the Bowman's capsule. Basically, all the blood components can be forced out here, except red blood cell and also protein, because the size is too big. Okay, this is number one, ultrafiltration. Number two is about the reabsorption. Because after the glomerulus, here we have the rear capillary surrounding the tube. And for the tube, first one is called proximal convoluted tubule. The U tube, this one U tube, this U tube here, is called loop of Henle. And then here is the distal. So combined is really PhD. Okay, proximal, Henle, and distal. PhD can be doctor, PhD, okay, or Pizza Hut delivery, PhD. So proximal, Henle, and distal. About this one, normally if come out in structure, let's say, they will give you a table. Okay, for example, when they give you the table one, they will show, let's say, okay, at the proximal, got glucose present. When come to the distal, oh, no more glucose present. Means the fluid, the filtrate fluid in the glomerulus, okay, sorry, in the proximal here, they actually got the glucose. When come to the distal, no more glucose. Explain why. Then answer is all the glucose is reabsorbed at the proximal convoluted tubule by active transport. So when come to distal, of course, no more sugar already. But how about special case? When they show, okay, glomerulus got sugar. When come to the distal, still got sugar. So like this is the abnormal case, then answer is the diabetes. Okay, yeah, for diabetes, kencing manis. Okay, so normally for this nephron come out, for structure or assay, they like to show you the table. Then from the table, they will show the sugar amino acid or maybe the urea in maybe proximal or in distal. Then compare the difference. You have to explain one. Okay, let's say why at the proximal got amino acid or sugar. Distal don't have. Because all the amino acid, all the sugar will be reabsorbed at the proximal by active transport. It's all. Serap semua, so it's through active transport. Okay, I hope this one can help you. Both also consider big topic. The blood pressure can go to assay. The nephron can go to assay and also structure. Okay, hope this one can help you. Okay, for the next one. Okay, so this one is about you're asking about the colonization and the succession process in the mangrove swamp area, Kawasan Payapakau. Okay, for this one, yes, it's for assay or can be for structure. Okay, like this. First, you must understand if for structure, they will give you the diagram. The mangrove swamp area diagram there. And then you can see also got the Avicennia, Soalan species. You can see the plant with the Puma torfol root system. The second one is the prop root. The second one normally is the rhizophora. And then number 3 is actually about the Bulgaria. Okay, so number 1, you must memorize the scientific names of these 3 plants. Because for the colonization, the pioneer species for the mangrove swamp area is Avicennia and Soalan. And then they will be replaced, means will be succession by the rhizophora. And then by the Bulgaria. So we try here. Okay, number 1 is the Avicennia. This is the first species, the pioneer species that come to the mangrove swamp area. One more is the Soalan species. Okay, for scientific name, you must underline the name standard. Then these two will be replaced by rhizophora. And then by, rhizophora, sorry, SP dot also a species. And then by the Bulgaria species. Okay, so from here, how to memorize the sequence? I memorize the Avicennia, Soalan, I put AS. And then by rhizophora, the R, and then by the B is the Bulgaria. So I memorize AS and then RB. I change to AS become the Asian, Asian. RB, I put it rabbit. So Asian rabbit, AS, RB. Okay, yeah. Of course, depends on its assay or its really structure here. If let's say for the structure, they will give you also the diagram. So from the diagram, sometimes they give you the root system with the diagram. So what happened is, you need to know, you have to recognize the diagram. This one, if you see the diagram, you can see Puma torfol only. So they ask about the plant. So 2 possible answer, Avicennia, Soalan. Okay, how come this one can be replaced by the rhizophora? How come the pioneer species Avicennia and the Soalan that can be replaced by the rhizophora, means succession? Okay, answer is this Avicennia and Soalan, they are actually plant, pokok. Okay, they will die one day. So after this plant die, they become the humus, means the fertilizer. They become the humus and then give to the soil. So the soil now with slightly more humus, and the land area will increase a little bit and become slightly more compact, not that soft anymore for the land, for the soil here. So become harder and also higher area. So with higher area, less sea water, less salty. So this new environment is more suitable for the one called rhizophora to survive here. to grow on here compared to the starting one, because starting one is more seawater, the muddy soil is very soft. That one is suitable for the Avicennia. So why only Avicennia, Soalan can be the pioneer species? Why only these two can colonize the mangrove swamp area, means the first place? Answer is, for the Avicennia and Soalan, you must understand, they got the Puma torfor. They got extensive underground cable root. So the underground cable root is to provide good support. Imagine, this is actually mangrove swamp area at the seaside or maybe at the riverside. The water is very strong one. So make sure that they can get very good support in order for them to grow there. So answer for both Avicennia and the Soalan, they got extensive cable root, underground cable root to provide good support is number one. Number two, for this root, if they grow underground, top here covered by the seawater, they cannot breathe at all. So they got very special system for the root, is all the way grow up. It's called the Puma torfor. Puma torfor here, the root is grow all the way up until above the seawater level. It's called Puma torfor. So for the Puma torfor, one more name is called the breathing root. So they can breathe. Can breathe means can inhale the oxygen for the root to perform active transport. Once got active transport here, of course, they can absorb the mineral from the seawater. They can absorb the salt from the seawater. Then only followed by osmosis, water can be absorbed by the root. The main point is, this Avicennia, they can become the pioneer species just because of they got a very good root system, number one for support, number two for breathing, the Puma torfor. Because when they can breathe, they can absorb the water from the seawater, they can survive here. They can colonize at this area. Plus all this mangrove swamp area, all the plant here also got thick cuticle. Why must be thick cuticle on the leaf? To reduce transpiration. Because it's actually the seaside, so under very hot sunlight one. So for this plant, they want to survive here, must have very thick cuticle to reduce the rate of transpiration. Okay, yeah, hope this one can help you understand slightly better. So for this one, is if to memorize, is a root system, scientific name, and the characteristic. Okay, so move on to the next one. Shahira, okay, from this student. Okay, what does lymphatic system do? Or means about the function of lymphatic system. I put it this way. Lymphatic system is also part of our transport system. We got actually two transport systems. The main one, of course, is our blood system. Then the secondary is the lymphatic system. So basically, lymphatic system, they got two main functions. One is for transport. Number two is for the defense. How come this system also involved in the defense system? Because if you can recall, lymphatic system got the structure is called the lymph node. The lymph node here, they can actually produce the lymphocyte. So they can produce lymphocyte, means can produce antibodies. So with the antibodies, they can actually kill the bacteria. So involved in defense system. So one of the function for this system is to filter bacteria, means to kill bacteria. How? By producing antibodies. Who produce antibodies? Lymphocyte. So lymphocyte is produced in the lymph node. Understand that? So the swollen part of the lymphatic vessel, that one is called the lymph node. So the lymph node there actually let the lymphocyte to become matured. So once matured, they can produce antibody to kill bacteria. This is number one. Number two, the main function is to return all the excess interstitial fluid from the intercellular space, okay, return to the blood circulatory system. I repeat, is to return, means to transport all the excess interstitial fluid all the way to the blood system. Let's say if our lymphatic vessel got blockage, all the excess, the 10%, all the excess interstitial fluid cannot be returned to the blood system. Means the excess fluid will accumulate outside the tissue here. Answer will cause edema, swollen. And the disease normally is called elephantiasis. Okay, so I repeat here, lymphatic system got two functions. One, answer is defense. Okay, with lymphocyte, can produce antibodies, can kill bacteria. One. The main function is to transport or to return the excess interstitial fluid to the blood system. If fail to return, answer is edema, means swollen, bengkak, like elephantiasis. Okay, hope this one can help you. So, two main function. If you want to add extra 1 point for lymphatic system, then it will be about the top of the lacteal, also under transport, because lacteal is the lymphatic capillary in the villus. They can transport lipid, transport the fatty acid, okay, return to the blood system. Okay, hope it can help you understand slightly better. Okay, next one. Okay, can you explain about mitosis and meiosis, the differences and other? Okay, so about mitosis, meiosis here. Okay, 1 minute. So both also under chapter cell division. Okay, is your Form 4 syllabus, chapter 5 here. Okay, mitosis, okay, basic info first. Mitosis can happen in the whole body. Every single cell, we can perform mitosis from top to bottom. We can grow taller, taller, and taller, is mitosis. So for growth, for cell repairing, answer is mitosis. Meiosis only got one function, is to form the gamete. So which part of the body can perform meiosis? Answer is sex organ. For boy, answer is testis. For girl, answer is ovary. So basically for this one, meiosis, only one purpose is to form gamete, means the sex cell, sperm or ovum. But for mitosis, is the whole body can perform for growth, for development, for cell repairing, for cell replacement. Okay, and then about the detailed one, for mitosis, okay, for mitosis, produce one cell, it will split to form two diploid cells, means one become two. But for meiosis, actually one become four, because mitosis only got one time cell division, but meiosis got two times cell division. So got meiosis 1 and meiosis 2. Okay, they will divide from one cell, split into two cells. Then from the two cells, split one more time, you will get total got 4 cells. So under actually the meiosis got 2 cell division and form 4 cells. But mitosis only split one time and form 2 cells. So 4 cells and 2 cells. And then for the meiosis, all also haploid. Mitosis, all diploid. Okay, so one more time here. Number 1, mitosis, the purpose is for growth, cell repairing, cell replacement. Meiosis is to form gamete. Then about the location, meiosis only sex organ can perform. Mitosis is the whole body. Number 2, okay, for meiosis can get 4 daughter cells. This one, mitosis only got 2. Meiosis come out is the haploid cells. Mitosis is diploid cell. Okay, so about the cell division, meiosis got 2 times cell division. Mitosis only 1 time cell division. Okay, one more point is mitosis is a continuous process. Means after the split through mitosis, the new cell can split again in future. It's a continuous process. But meiosis, no, only one cycle, then stop. So basically, if as I come out about the cell cycle can repeat only for mitosis, because mitosis is actually continuous process. But meiosis only one round and then stop. Okay, hope this one can help you understand better about the differences between mitosis and meiosis. Okay, so we come to the next question. Okay, for the next one is about the caffeine. Is it dangerous if we drink caffeine frequently? Caffeine means something like coffee or tea, and is related to neurotransmitter. Okay, so basically number 1, you must understand caffeine. Of course, in tea, in coffee, they've got the caffeine. So caffeine is a type of stimulant. It's under the group called stimulant drug. Yes, it's a drug, it's stimulant, but if you drink, it's okay, you won't get any hallucination, it's okay one. So basically caffeine, the effect is increase your heartbeat. Okay, let's say for some people, they drink coffee every day. Okay, so drink coffee every day. Is it good or bad thing? Depends on your habit. Let's say if you are the one that, okay, morning, you must take a cup of coffee or tea for you to continue your day, then it's okay. Because caffeine is just like increase your heartbeat. Of course, also increase your heartbeat means can increase the impulse flow faster. Then got more neurotransmitter there, so increase the impulse flow, so increase your heartbeat is a stimulant, so make you like feel more alert, okay? And then for the caffeine, any bad effect or not, dangerous or not? So answer is if one day you drink only 1 to 2 cup coffee, so it's okay, okay? Just that it's about the timing that you drink. Number one, if you drink in morning time, it's all fine. Afternoon is still good, just that it's not recommended you drink something with caffeine at night. Because after you drink the caffeine, you feel alert. You cannot sleep already. So when you can't sleep, will affect your body health. Okay, yeah. So basically, caffeine you can drink, even drink every day is also can if you want to. Okay, just that don't drink it at night because it will affect your sleeping cycle. And one more bad effect about caffeine is may cause dehydration. When you drink too much caffeine, your skin become a little bit dry because normally after you drink coffee or tea, the pure one, then you feel like you want to go to pee more frequent because caffeine got one function can link to the impulse one, so it can link to the hormone one. Actually, caffeine can inhibit the release of the ADH hormone. The hormone that can control the water reabsorption from the kidney. So the more caffeine you put in, so you release less ADH. Means you will pee more. When you pee more water come out. So what you can do is drink more water. Okay, yeah. So for the normal coffee you can just drink is okay, but not at night. Okay, number one. And in this world also got many things also got caffeine one. So depends on how much that you take the level. Try not to be over because it will affect your water, the body water. So become dehydrated. And for some people, when the caffeine is too strong, the heart beat too fast. You feel not comfortable. You feel maybe a bit pain one. So if like that, then better don't take the caffeine. But if you think you are okay, your coffee with any tea or coffee, just like it, then just drink. Okay, yeah. But daytime, not at night. Okay. Okay, so next one, we come to this student from CAT also. So please explain fight or flight situation. Okay, for fight or flight situation like this, okay, let's say when you encounter an emergency situation, let's say suddenly someone want to attack you. Okay, so within the 1-2 second, you have to respond very fast. Either you want to fight back or you want to run away. It's called flight. So it's called fight or flight, means run away, terbang. Okay, yeah. So for both, you need extra energy to perform. Let's say you want to fight, you need energy to fight. You want to run away fast, you also need the extra energy one. So from where can get the extra energy? Answer is the hormone called adrenaline hormone. So for this one, you must mention the hormone that involved is called adrenaline hormone. They're produced or secreted by the adrenal gland. Okay, let's say when human we feel nervous, we feel excited, we feel angry, or we feel it's emergency. Okay, so from here, from the feeling here, we send the impulse to the adrenal gland, the medulla layer. Okay, so secrete more adrenaline hormone. Okay, this hormone, the function will increase your heartbeat. Okay, increase your heartbeat means we send impulse to increase your heartbeat one, and then we also increase your breathing. So once can increase your breathing, like feel like breathe in and out faster and deeper. When you breathe in faster and deeper, you inhale more oxygen. The heart also beat faster, right? So can pump more oxygen to the brain for you to think faster, and pump more oxygen and sugar to your body muscle for you to respond faster. So that, okay, in this emergency situation, you have to decide very fast. You want to really fight back or you want to run away. So for both also you need energy. So for this case, what you need is the hormone, main point is adrenaline hormone. Who produce or which gland produce this hormone? Answer is adrenal gland. Okay, yeah. Effect of this hormone: increase heartbeat, increase your breathing, can also increase the conversion from glycogen to sugar. Because you want to burn more sugar, right? You must get more sugar. From where can get more sugar? Answer is glycogen. So this one called adrenaline got the ability to convert glycogen back to sugar. Then plus the heart pump faster, right? So pump more sugar plus pump more oxygen to the muscle, to the brain, to the whole body for to respond faster. Okay, yeah. Okay, this one fight or flight situation is very good one because can link impulse all the way to the hormone, and the hormone must be adrenaline hormone. Okay, yeah, and then talk about the effect. Explain about the effect like increase heartbeat, increase breathing, increase the conversion of glycogen to glucose, increase the sugar level, increase your metabolism for to get more energy to fight back. Okay, yeah. Hope this one can help you about fight or flight situation out before in SPM essay and give total about 10 marks essay. You can refer also from the past year paper. So basically the main info is adrenaline and the effect. Okay. Okay, the next soalan from Kiki Lala, betul ke stres boleh aktifkan sel kanser? Tak semestinya. Okay. Ya, setiap orang pun adanya stres kan. Katakan untuk belajar, kamu punya stres datang daripada mungkin ialah result daripada exam kamu. Okay, stres tak semestinya boleh mengaktifkan sel kanser, tapi stres boleh menyebabkan kamu bebaskan hormon adrenaline. Okay, so yang tadi punya about fight or flight response ialah daripada stres. When you feel stress kan, heartbeat lebih cepat. When you feel stress kan, you breathe, bernafas lebih cepat juga. So basically stress is about fight or flight response. Bukan tentang kanser punya. Ini tak ada kaitan. Okay, kalau stress boleh aktifkan sel kanser, saya rasanya semua orang di city pun ada kanser. Tadi tak ada punya. Jadi stress boleh tak aktifkan sel kanser? Ini tak ada kaitan. Kanser ialah disebabkan adanya mutasi berlaku. Mutasi daripada katakan radiasi, daripada UV, daripada mutation, bahan kimia yang tak bagus, preservatif, itu boleh aktifkan sel kanser. Tapi stres bukan. Stres tidak, tak kira punya. Kalau stres dia lebih ke bahagian a bit psycho problem, stress. So basically, you have to find a way to reduce your stress level. Even as a student, now the student also very stressed kan. Exam is coming around the corner. So for you, SPM another less than 1 month. So basically, you feel very stressed. What you can do is listen to music, okay, make your mood feel like lebih bagus, lebih selesa. Okay, yeah. So stress one link to cancer one, don't worry. Okay. Okay, next one. Okay, how to know whether the type of solution is hyper, iso, or hypo? The percentage used for the solution as well. Okay, senang saja. Untuk bab ini, kalau nak keluar, dia akan bagi diagram. Okay, akan bagi diagramnya. Daripada diagram sana, you boleh tahu adakah itu larutan, the solution is hypo, hyper, or isotonic. Let's say they give you about the plant cell. Okay, give you plant cell. Okay, example. Here got 3 plant cells. Okay, ini okay, this one immersed in solution A, solution B, and solution C. Okay, so now in 3 different solutions for the plant cell A, B, C here, in the solution A, solution B, and solution C here. They say separate one, separate. Okay, so from here, solution C, very obvious is the hypertonic. No matter what percentage you give you, once with the diagram like this, confirm is hypertonic. Hyper is actually concentrated. Let's say it's about 30%, 30. Salt solution. So it's considered concentrated here. So water will diffuse out, and you can see got plasmolysis here. So it's hypertonic. Compare A and B. Look at the size of the vacuole. A got bigger vacuole, and B is smaller vacuole. So when it comes to the smaller vacuole and the bigger vacuole, means this one is water all the way diffuse in. Diffuse in means this is the hypotonic. Hypo means dilute. Let's say only 1% NaCl solution, then is hypo, dilute, or maybe it's pure distilled water, pure water also dilute. Then this one is the in between, then must be the isotonic. Don't worry, for SPM exam, they won't ask you just state the percentage of the solution direct. They will give you the diagram. From the diagram, you can actually estimate which one is hypo, hyper, and the isotonic. Okay, okay. Unless they ask about human more specific, let's say for the human red blood cell, it's about 0.9% NaCl is the iso. So above 0.9 is hyper for the salt solution, below is the hypo. Okay, and normally this one come out with the diagram. Okay, with the diagram, don't worry. So based on the diagram, you can roughly know that is it normal, means iso, or the vacuole expand. Let's say for plant cell, vacuole expand, then it's the hypotonic solution, or the vacuole become very small, string and the membrane move away, means it's plasmolysis, then answer is the hypertonic. Okay, Okay, hope this one can help you understand slightly better about the iso, hypo, and hyper. Unless they come out, it's not the plant cell, it's the red blood cell. If for red blood cell, will be the same thing. Look at the red blood cell. Is it normal? Normal, then it's iso. If the red blood cell bursts, then it's in the hypo. If the red blood cell shrink, it's in the hypertonic. Okay? Okay, so we move on to the next one. From another student. Hemi. Okay, so explain about antigen A and B. Okay. This one is the Form 5 Chapter 5. Form 5 Chapter 5 about the antigen A, B, about the blood type here. Form 5 Chapter 5 and Form 5 Chapter 1 also. Okay, for the antigen A and antigen B here is about the red blood cell. Okay, let's say for human, we have 4 different types of blood groups or blood types. We have the blood type A, we have the blood type B, we have the blood type AB, and the blood type O. So let's say if on the surface of your red blood cell, okay, for the red blood cell, you actually have the antigen type A, then answer, you have blood type A. If you got antigen type B on your red blood cell, you have blood type B. You got both A and B on your red blood cell, then you have blood type AB. How about you don't have A and B, you have no A, no B, nothing, it's the blood type O. Okay, okay, one more time here. For red blood cell, we talk about antigen A and the antigen B. If only got antigen A present, it's called blood type A. If only B present, it's called blood type B. Both also present, blood type AB. Don't have A, don't have B, nothing there, is the blood type O. Okay, so this is the one that determine our blood type. Why our blood type, some got A, some people B, some people C, because of this one only. Okay, hope this one can help you understand slightly better about the antigen. Okay, okay, so for the blood type, okay, I can explain a bit more for you. Actually, for those blood type A, beside the antigen A, you have to know, also we have something called the antibody in the blood plasma. Let's say I have blood type A. So actually I have the antigen A on the surface of my red blood cell. So if I got the antigen A on my red blood cell, then in my blood plasma, I can only produce the B antibody. Again, if I'm blood type A, okay, my red blood cell got the A antigen. Then in my blood plasma, I can only produce the B antibody. Make sure that they are not complementary. If I got blood type A, I got the A antigen, and let's say I produce also the A antibody. So it's like I produce antibody to kill myself. It's impossible, like commit suicide. Okay, yeah, so no. If I have A antigen, I can only produce the B antibody. Understand or not? So that there's no attack. So everything is okay, I can survive. If I have the B antigen, I can only produce the A antibody. I got both A and B antigen, I don't form AB antibody, just terbalik. So that everything is okay. For this reason, when it comes to the blood transfusion, that's why A and B, they cannot mix. Because the antibody will attack the antigen. When A, blood type A, and the blood type B, they come to transfusion, they mix. So blood type A and B cannot mix because of the antibody antigen reaction. Okay? Okay, hope this one can help you understand slightly better about the blood type. And then about the blood type also got the Mendel thing. You have to know that how to do the calculation, the Mendel's formula. Let's say someone blood type O, married with someone blood type A. So what's the possible answer for their next generation? The percentage, let's say one blood type A, one blood type O. So can they get blood type A or not? Yes or no? Can they get blood type O or not? Yes or no? And what's the percentage? It's about the calculation one, okay? And also about the blood transfusion, about the ABO blood type. Okay, yeah. So basically, I hope that today you can really benefit from this program that can help you in your coming SPM. Okay, so we're going to close here. So thank you for watching, and don't forget to watch the recorded section of TanyaCikgu and other videos at spmflix.com. So TanyaCikgu will be live again on 18, 19 and 20th October to answer more of your questions. And bye. Thank you.